Okay, so you've got this hyperstabilized mRNA message producing spike proteins. Oh, and by the way, a lot of other things. Indefinitely.
There's no study showing when it stops making them.
Right. So we've seen that they persist for a long time, and the end of our ability to see that is the end of how long it's been studied. So
Correct.
Who knows how long it lasts. I think of it as like a fiberglass log in a zoo exhibit, right? It looks like a log, and you would think the termites and saprophytic fungi would eventually take it apart, but it's fiberglass. No, it's sticking around.
Great analogy.
Indefinitely. But anyway, okay, so you've got the ribosomes in these cells producing a huge amount of product, some of which is presumably spike protein or spike protein-like. Mm-hmm. Some of it's not because of frameshift issues.
Bingo.
And so you've got a lot of garbage protein, right? Just like, monkeys at a typewriter protein. That is going to start crowding a cell.
Well, yeah, so it's going to cause internal cellular damage. And that cell now has two options. One is to self-implode, autophagy.
Yep. Right.
Or two,
T cells.
It's going to, a T cell can come blow it up, or it can try to adapt, and it may have a maladaptive process to the buildup within it, which may lead to mutation.
Yeah.
And the other thing to consider is there are multiple cell cycle pathways and multiple different proteins that our body makes to stave off cancer. We have a whole family of genes called P53.
Yep.
It's a tumor suppressor family of genes. It's not just one gene, it's a tumor suppressor family. The spike protein binds to the P53 receptor. It does.
Yeah.
And think of the other, we don't even know what the shorter aberrant proteins, they may bind as well. So now you have this whole milieu of different proteins that can bind there. We know it binds to the BRCA gene in silico. So we have known cancer regulatory genes that can be bound and dysregulated by this protein made by this synthetic shot. Now, in addition, and this is critically important, if you look at Moderna's patents, they were highly concerned about DNA being in a product because it could lead, and I have this in some of my talks, and if you want to put it on the website, I'll send it to you. Moderna's on patent because DNA in the shot can lead to mutation and carcinogenesis. In the BioNTech and the Pfizer applications, they said, well, this is lipids and mRNA. We're not even going to look for mutation and carcinogenicity because it's mRNA. Well, it's not, because what we found is in multiple laboratories around the world, there is contaminating DNA within these injected products, which also have sequences in them which help them co-locate into the nucleus of the cell. They are hyper designed to go where you don't want your mechanisms messed with.
You're talking about SV40.
SV40 promoter region. And
so SV is simian virus.
Number 40
this is
from a green monkey.
Yeah, it's a biotech promoter region, something that came from a green monkey that is now utilized in, people may have heard us talk about there having been an initial process for the production of these shots that did not involve DNA plasmids, and then there was a second process which did not get safety tested.
Such an important story.
Yeah, it's a really important. I mean, it's a bait and switch. Right?
It's an absolute bait and switch. They did a synthetic thing that they used for the trial, and then the population that got the shot got process number 2, which was the contaminated process,
which has all of this DNA,
all the junk in it.
It's got a tremendous range in lengths, so basically you're getting an arbitrary set of DNA, but some of it includes this SV40 promoter, which is strongly likely to result in integration in the nuclear genome.
And that's been shown by Dr. Alden in liver cell lines. Dr. McKernan, last week, he showed it in an ovarian cancer cell line integration.
Yeah.
And there are 2 other more obscure papers that show integration as well. So there are 4 papers now that show integration events.
All right. Now, if I can flash us back to our recent meeting in Romania, I had a moment of great pride that came through you, where you gave a talk and you described the fact of cancers that had been apparently induced by these mRNA vaccinations, or so-called vaccinations. And you said, kind of offhandedly, as you showed a slide in which they had been labeled with something that allowed you to see it, you said that the cancer cells were producing spike protein. And I thought, what did he just say? Mm-hmm. Because that implies, in order for that to be true, a cancer starts with a single cell that becomes dysregulated. In order for all of the daughter cells that are produced in that cancer to be producing spike protein, that spike protein has to be carried along and duplicated, which suggests integration.
Every time that cell divides, then that next cell is still expressing that sequence to make that protein, and then we put a little tag to bind that protein to show that it's there. And you're one of the few people that picked up on that. There's only a handful of people. When I showed that slide, Dr. Burkhardt, the late great Dr. Burkhardt, he showed it in a gastric cancer, the lymphoid cells of a gastric cancer. So every lymphoid cell had expression of spike protein. Now, I showed it in a pancreatic cancer when I was doing testing in my laboratory. That says that either it's integrated into the DNA, but it can also co-locate into the nucleus and still be viable if it's red. Mm-hmm. But something's happening in order for that spike to still be expressed each time that cell divides.
Well, you would get a pattern, a rapid pattern of decrease in the production of spike protein if it was not being duplicated somehow. So something is duplicating that mRNA message or integrating it into the DNA in some way, which involves a set of pathways that really shouldn't be operating.
Correct.
So anyway, fascinating and terrifying.